Welcome to Lecture 3 of Chapter 21, Lipid Biosynthesis. This lecture will focus on the biosynthesis and transport of cholesterols, steroids, and isoprenoids. We will begin this chapter with an account of the main steps in the biosynthesis of cholesterol from acetate, and then discuss the transport of cholesterol in blood and its uptake by cells. In addition, we'll also look at normal regulation of cholesterol biosynthesis. Finally, we'll consider some cellular components that are derived from cholesterol, such as bile acids in the liver and other steroid hormones. Cholesterol, steroids, and isoprenes are compounds that are chemically related and distinct from triacylglycerols, phospholipids, sphingolipids, and plasmalogens. Cholesterol is very well known for its role in human cardiovascular diseases. However, it is less known that cholesterol's crucial role as a component of cellular membranes and as a precursor of various steroid hormones and bile acids. Cholesterol is an essential molecule in many animals, including humans, but it is not required in the mammalian diet. The reason is that all cells can synthesize cholesterol from its simple precursors, such as isoprene. Isoprene is the basic building block of all steroids and cholesterols. This slide describes a brief overview of eukaryotic cholesterol biosynthesis. Synthesis of cholesterol takes place in 4 stages, as shown in this figure. In the first stage, condensation of 3 acetate units results in the formation of a 6-carbon intermediate, mevalonate. Conversion of mevalonate to activated isoprene units happens in the second stage. The activated isoprene is a 5-carbon unit. In the third stage, polymerization of 6 5-carbon isoprene units results in the formation of a 30-carbon linear molecule called as squalene. And in the fourth stage, cyclization of squalene results in the formation of 4 rings of the steroid nucleus, with further series of changes such as oxidation, removal or migration of methyl groups to produce cholesterol. Let us look at the first step in the biosynthesis of cholesterol. This step involves the formation of mevalonate from acetyl-CoA. 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA, which further condenses with a 3rd molecule of acetyl-CoA to yield the 6-carbon beta-hydroxy beta-methyl glutaryl-CoA or HMG-CoA. The first reaction is catalyzed by the enzyme acetyl-CoA acetyltransferase. The second enzyme, which may be familiar to you, is HMG-CoA synthase. Remember, HMG-CoA synthase is also involved in ketone body synthesis. But the cytosolic HMG-CoA synthase in this pathway is distinct from the mitochondrial isozyme that catalyzes HMG-CoA synthesis in ketone body formation. We have seen this before in previous chapter. The third reaction, that is a conversion of HMG-CoA to mevalonate, is the committed step. This involves reduction of HMG-CoA to mevalonate. And in this process, 2 NADPH molecules gets oxidized to NADP. HMG-CoA reductase catalyzes this reaction. HMG-CoA reductase is an integral membrane protein of smooth endoplasmic reticulum. and is the major point of regulation on the pathway to cholesterol. Step 2 of cholesterol biosynthesis involves conversion of mevalonate to activated isoprene units, as shown here. So how does mevalonate get converted to isoprene units? In the first step, mevalonate is converted to 5-phosphomevalonate by the action of the enzyme mevalonate 5-phosphotransferase, which uses ATP as a cofactor. In the second step, 5-phosphomevalonate is further phosphorylated by phosphomevalonate kinase, which converts 5-phosphomevalonate to 5-pyrophosphomevalonate. In the third and the crucial step, 5-pyrophosphomevalonate is converted to delta-3 isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. And this is catalyzed by pyrophosphomevalonate decarboxylase. Now this enzyme does two reactions at once. The first reaction is activation of this hydroxyl group using ATP. So it literally phosphorylates this hydroxy to produce 3-phospho-5-pyrophosphomevalonate. In the second step, it decarboxylates this carboxyl group. So how does the decarboxylation mechanism goes? So in this case, the electrons are pushed from here to here, and this bond pushes electrons here, and this phosphorylated hydroxy then leaves. So the mechanism is quite simple. As a result, you end up with delta-3 isopentenyl pyrophosphate. Now, delta-3 isopentenyl pyrophosphate, or IPP, can further isomerize to form dimethyl allyl pyrophosphate, or DMAPP. And the isomerization reaction is where this hydrogen moves here, and this double bond abstracts a proton. Step 3 in the biosynthesis of cholesterol involves condensation of 6 activated isoprene units to form squalene. In the first step in the synthesis of squalene, 2 activated isoprene units, dimethyl allyl pyrophosphate and delta-3 isopentenyl pyrophosphate, condense together in a head-to-tail fashion, displacing one set of diphosphates, forming a 10-carbon geranyl pyrophosphate molecule. The head is the end to which pyrophosphate is joined. This is the tail. Geranyl pyrophosphate undergoes another head-to-tail condensation with isopentenyl pyrophosphate, yielding the 15-carbon farnesyl pyrophosphate. Finally, 2 molecules of farnesyl pyrophosphate join head-to-head with the elimination of both pyrophosphate molecules, or both pyrophosphate groups, to form squalene. Squalene has 30 carbons, 24 in the main chain, and 6 of them, 1, 2, 3, 4, 5, 6, in the form of methyl group branches. Step 4 involves the conversion of squalene to cholesterol, which has a 4-ring steroid nucleus. The relationship of squalene, which has a linear structure, to the cyclic structure of sterols, just cholesterol, is apparent from the way squalene is represented here in this figure. All sterols have 4 fused rings, as shown here, 1, 2, 3, 4. And they also have a hydroxy group at C3 position, and hence the name sterols. In the conversion of squalene to cholesterol, the first step involves the oxidation of squalene to squalene 2,3 epoxide. and this is catalyzed by the enzyme squalene monooxygenase. This enzyme adds one oxygen atom from O2 to the end of squalene molecule to form an epoxide, that is squalene 2,3 epoxide. NADH then converts the other oxygen atom to a water molecule. Squalene 2,3 epoxide is positioned in such a way that a remarkable concerted reaction can convert the linear squalene epoxide to a cyclic structure. You do not have to understand as to how this concerted reaction happens. But in animal cells, this cyclization results in the formation of lanosterol. Now, as you could see, there is a carbocation intermediate that is formed. So this concerted reaction goes through a carbocation intermediate. In case of plants, squalene 2,3 epoxide is converted to stigmasterol, and in fungi to ergosterol. Lanosterol in animals is further converted to cholesterol in a series of steps. It's about 20 reactions is involved in converting lanosterol to cholesterol. So, the cholesterol biosynthetic pathway is one of the most complex and extraordinary pathway that is known. Cholesterol has several fates. Most of the cholesterol synthesis in vertebrates takes place in the liver. A small fraction of the cholesterol made in the liver is incorporated into the hepatocyte membranes. But a majority of these cholesterols synthesized are exported as bile acids, biliary cholesterol, or cholesterol esters. Bile acids, which is one of the three forms of cholesterol exported from the liver, is stored in the gallbladder. Bile acids is the principal component of bile, which is a darkish green or yellowish brown fluid that is stored in the gallbladder and secreted into the small intestine after a fatty meal. Bile acids such as taurocholic acids serve as emulsifiers in the intestine specifically, and this is to convert large particles of fats into tiny micelles and thereby greatly increasing the surface at which digestive lipases can act upon. Bile also contains much smaller amount of cholesterol or biliary cholesterol. In other tissues, cholesterol is converted to steroid hormones. This figure shows the various structural differences in molecules other than cholesterol. Like I said, cholesterol is converted to other steroid hormones in tissues other than liver. In liver, cholesterol is converted to bile acids. Bile acids have this structure. Cholesterol is also converted to cholesterol esters in the liver through the action of acyl-CoA cholesterol acyltransferase or ACAT. This enzyme transfers a fatty acid group from fatty acyl-CoA to the hydroxyl group of cholesterol. What this does is converts cholesterol to a more hydrophobic cholesterol ester and prevents entering of cholesterol into the membranes. Cholesterol esters have two fates. They are either transported in secreted lipoprotein particles to other tissues that uses cholesterol, or they are stored in the liver in lipid droplets. Cholesterol and other lipid particles are carried on lipoprotein particles. The reason is that cholesterol and cholesterol esters like triacylglycerols and phospholipids are essentially insoluble in water, and they must be carried in lipoprotein particles from one tissue to another. In blood plasma, plasma lipoproteins carries cholesterols and other lipids. These are spherical particles. Apolipoproteins are proteins in its lipid-free form combined with lipids to form several classes of lipoprotein particles. These spherical complexes have hydrophobic core, which has cholesterols, triacylglycerols, and cholesterol esters stored in them. On the outside, the protein has hydrophilic amino acids and also a phospholipid monolayer. Different combinations of lipids and proteins produce lipoprotein particles of varying densities, ranging from kilomicrons to high density lipoproteins. There are four major classes of lipoprotein particles, and these particles can be separated by ultracentrifugation, and their composition varies between class of lipoprotein. For example, kilomicrons is the least density. That is because of the least protein content in them. But kilomicrons has the highest amount of triacylglycerols. High density lipoproteins, as the name suggests, has the highest density, and that is because of the amount of protein these particles have. On the contrary, high density lipoproteins have the least triacylglycerol contents. Low density lipoproteins has the highest cholesterol and cholesterol ester content in them. The four classes of lipoproteins can be visualized in an electron microscope after negative staining. On the top left is kilomicrons, that has about 50 to 200 nanometers in diameter. On the top right is the very low density lipoproteins that has a diameter of 28 to 70 nanometers. On the bottom left is low density lipoproteins, and they have a diameter of 20 to 25 nanometers. And the smallest is on the bottom right, that is high density lipoproteins that has a diameter of 8 to 11 nanometers. Shown in this slide is a table depicting the identities of 10 distinct apolipoproteins that are associated with various lipoprotein classes. Each class of lipoprotein has a specific function determined by its synthesis, lipid composition, and apolipoprotein content. These apolipoprotein contents act as signals. They also target lipoproteins to specific tissues or activate specific enzymes that act on lipoproteins. These proteins have also been implicated on various diseases. We will talk about the function of at least one of these apolipoprotein in the coming section. For example, ApoB48 is an apolipoprotein that is associated with kilomicrons. And this is a humongous protein. It has a molecular weight of 242,000 Daltons. And its function is in cholesterol transport and clearance. Next, let us consider the biological roles of these four classes of lipoproteins in trafficking cholesterol and triacylglycerols. The biological roles is shown in this pathway. And this pathway is divided into two parts: exogenous pathway and an endogenous pathway. The exogenous pathway involves the trafficking of triacylglycerols that are formed in the diet to the liver. The endogenous pathway involves the formation of very low density lipoprotein in the liver that exits it and enters back as low density lipoprotein. Let us first consider the exogenous pathway. Kilomicrons are synthesized from dietary fats, and it happens in the endoplasmic reticulum of enterocytes, the cells that line small intestine. Kilomicrons, after getting synthesized, move into the lymphatic system and enter the bloodstream through the left subclavian vein. The apolipoproteins of chylomicron include ApoB48, the one that we talked about in the previous slide, ApoC2, and ApoE. ApoC2 activates lipoprotein lipase in capillaries of adipose tissues, muscle, and mammary gland. Once lipoprotein lipase is activated, it cleaves free fatty acids from triacylglycerols contained in chylomicrons. These free fatty acids enters all these tissues: adipose, the muscle, and the mammary glands. Once this happens, chylomicrons are left with depleted triacylglycerols, some cholesterol, ApoE, and ApoB48. The remnants of chylomicrons then move through the bloodstream and enter the liver. Receptors in the liver bind to ApoE in the chylomicron remnants and mediate uptake of these remnants by endocytosis. So remnants of chylomicron enter liver via endocytosis. In the liver, remnants release their cholesterol and are degraded in lysosomes. This pathway, like I said, is the exogenous pathway. Next, let us look at the endogenous pathway. When the diet contains more fatty acids and cholesterol than are needed immediately as fuel or precursors to other molecules, they are converted to triacylglycerols or cholesterol esters in the liver and packaged with specific apolipoproteins in VLDL, or very low density lipoprotein. Excess carbohydrate in the diet can also be converted to triacylglycerols in the liver and exported as VLDL. In addition to triacylglycerols and cholesterol esters, VLDL contains apolipoproteins. The list is given in table 21.2. VLDL is transported in the blood from the liver to muscle and adipose tissues. In the capillaries of these tissues, ApoC2, remember ApoC2, ApoC2 activates lipoprotein lipase, which catalyzes the release of free fatty acids into the adipose tissues and muscle tissues. Adipocytes take up these fatty acids and reconvert them to triacylglycerols and store the products in intracellular lipid droplets. Myocytes are the muscle cells. In contrast to the adipocytes, primarily oxidize the fatty acids that are delivered from VLDL to supply energy. When the insulin level is high after a meal, the very low density lipoprotein serves primarily to convey lipids or triacylglycerols to adipose tissue for storage. Whereas in case of fasting, VLDL transports the fatty acids to muscle to convert them to energy. The loss of triacylglycerols to adipose or muscle cells converts some VLDL to VLDL remnants. And these VLDL remnants are called as intermediate density lipoproteins, or IDL. Further removal of triacylglycerols from IDL produces LDL, or low density lipoproteins. LDL is rich in cholesterol and cholesterol esters, and it also has ApoB100 as the major apolipoprotein attached to it. LDL carries cholesterol to extrahepatic tissues such as muscle and adipose tissues. Remember, VLDL carries triacylglycerols and transport them to various tissues, whereas LDL carries cholesterol and carries them to various tissues. Now, these tissues have plasma membrane LDL receptors that recognizes ApoB100 and mediate uptake of cholesterol and cholesterol esters. And that is how cholesterol from liver gets to other tissues. LDL also delivers cholesterols to macrophages, sometimes converting them into foam cells. We'll talk about foam cells in a little bit. LDL that is not taken up by peripheral tissues and cells return to the liver. So the transport goes through here, the red line here, returns to liver and taken up by LDL receptors in the hepatocyte plasma membrane. Cholesterol that enters hepatocytes by this path may be incorporated into the membranes or converted to bile acids or reesterified into cholesterol esters for storage within cytosolic lipid droplets. This whole pathway from VLDL to IDL to LDL and back to the liver is called as the endogenous pathway. Accumulation of excess intracellular cholesterol is prevented by reducing the rate of cholesterol synthesis when sufficient cholesterol is available from LDL in the blood. Cholesterol esters enter cells by receptor-mediated endocytosis. Each LDL particle in the bloodstream contains ApoB100, which is recognized by LDL receptors present in the plasma membranes of cells that needs to take up cholesterol. LDL receptors are synthesized in the endoplasmic reticulum and transported to plasma membrane after modification in the Golgi complex. At the plasma membrane, LDL receptors are available to bind ApoB100. Binding of LDL to an LDL receptors via ApoB100 initiates endocytosis. As a result of endocytosis, LDL is internalized in endosomes. The receptor containing portions of the endosome membrane are separated and these receptors are transported back to the plasma membrane for further binding of other LDL particles. The endosomes with LDL particles then fuse with lysosome. Lysosome contains lytic enzymes that degrade ApoB100, the proteins, cholesterol esters, thus releasing amino acids, fatty acids, and cholesterols. ApoB100 is also present in very low density lipoproteins. The only difference is that the ApoB100 in VLDL does not have its receptor binding domain exposed for binding to the receptors. Only when VLDL gets converted to an LDL particle does this receptor binding domain of ApoB100 gets exposed and can be recognized by LDL receptors. And that is how cells differentiate binding of LDL and VLDL. There are at least two genetic disorders that are related to cholesterol transport. Familial hypercholesterolemia have mutations in the LDL receptors that prevent the normal uptake of LDL by liver and peripheral tissues. The result of defective LDL uptake is very high blood levels of LDL and as a result, high levels of cholesterol. Individuals with this particular disorder have a greatly increased probability of developing atherosclerosis, a disease of the cardiovascular system in which blood vessels are occluded by cholesterol-rich plaques. The second disorder is Niemann-Pick type C or NPC disease. This is an inherited defect in lipid storage. In this disorder, cholesterol is not transported out of the lysosomes and instead accumulates in lysosomes of liver, brain, and lungs. This results in early death. NPC is a result of a mutation in either of these two genes, NPC1 or NPC2. These two genes are essential for cholesterol mobilization out of lysosomes and into the cytosol, where it can be further metabolized. These proteins, NPC1 and NPC2, act in tandem to transfer cholesterol out of the lysosome and into the cytosol for further processing of cholesterol metabolism. HDL, or high density lipoprotein, is the fourth major lipoprotein in mammals. HDL originates in the liver and small intestine and contains relatively little cholesterol and no cholesterol esters. HDL also contains ApoA1 as its primary apolipoprotein. It also contains enzyme lecithin cholesterol acyltransferase, or LCAT, which catalyzes the formation of cholesterol esters from phosphatidylcholine and cholesterol. LCAT on the surface of nascent HDL particle converts the cholesterol and phosphatidylcholine of chylomicron and VLDL remnants encountered in the bloodstream to cholesterol esters. This means that HDL is a cholesterol scavenger. It scavenges the excess cholesterol that is present in these chylomicron remnants and VLDL remnants. Nascent HDL can also pick up cholesterol from cholesterol-rich extrahepatic cells such as macrophages or foam cells. And as a result, this HDL is then returned to the liver, where the cholesterol is unloaded via the scavenger receptor SRB1. HDL thus carries out a reverse cholesterol transport.